Welcome back to the virtual classroom. We were talking about the Entamoeba Hystolytica in the previous lecture. In that we have seen what are the geographical distribution of Entamoeba Hystolytica, what are the habit and habitats of the Entamoeba. In that we have seen that these Entamoeba Hystolytica, they basically present in the large intestine, not in the small intestine. The reason is that we have seen that they require a neutral environment whose pH will be somewhat 6.5 to 7.5, and as the small intestine have alkaline pH, so they don't survive in that. They goes up to the end into the large intestine, and in that also we have seen different forms, magna and minuta form, and after that the third form, that is the cyst, encysted form. Now that thing was divided into three parts. One was the pre-cyst, one was cyst, and then metacyst. Okay, metacyst. Once this is formed, then through the fecal matter they passed out of the body and infect other host and try to enter other host by some kind of contamination, fecal contamination rather. Now in today's lecture we will talk about the life cycle, how the infestation takes place and what happens after that, we will see. So if we talk about the life cycle of Entamoeba Hystolytica, now we will go step by step what happens. So once, with the help of some kind of infection, means the fecal matter may get mixed with water or some food because of some reason. Because of that, the metacyst part, we have already seen the characteristic of metacyst. 4 nucleus is present in the metacyst. Okay, so this trophozoite, most of its life as a trophozoite in the body of human. Okay, so initially how the life cycle starts, through the contaminated food it enters into the body, which is the infective stage. This is the infective stage, metacyst. Once inside the body, through water or food, first it will be present in the stomach. Because of its cyst wall, a thick wall, nothing will happen to it, it is protected. It gets transferred into the next part, that is the small intestine. Now once it travels the small intestine, and from the small intestine it reaches the lower ileum. Remember, intestine is divided, small and large intestine, even small intestine is also divided. In that, the lower ileum or cecum part, where it escapes from the cyst wall, means up to this region, up to the lower ileum or the cecum, it is still in the metacyst stage. It is not coming out because it cannot sustain or it cannot live in the highly alkaline or highly acidic region. It has to come to the area where somewhat neutral part will be present, neutral pH will be present. Okay, so here at this position, ileum or cecum, it escapes from the cyst wall. So it is present, it is covered with the cyst wall. Now this process of coming out of the cyst wall is called as excistation. Okay, and it happens at the end of the small intestine, generally happens at the end of the small intestine. Now in this excistation, how it works exactly, how the perforation, what it does, it forms the perforation in the cyst wall, and from that perforated area it tries to come out, means pseudopodia are formed. These exert pressure on the cyst wall at the various points, and they've tried to find out the weak spot in the cyst wall, and with a small pore development takes place in the wall, and through which the tetranucleated form squeeze out of the cyst wall. As you can see in stage 2, as it is given in the diagram, that through the cyst wall a small pore formation will be there, and there it squeeze out of the cyst wall. So exactly how the perforation is made, it is still not clear, but amoeba probably secretes some kind of local cystolytic substances, definitely. Few authors have implicated that the enzyme trypsin in digestion of the cyst wall takes place, so they may secrete the trypsin, but excitation does not occur in the region of active digestion. Remember, that's why not in the stomach and not in the actual small intestine, but the end of the small intestine, at the ileum region, actually they come out, because once they are out, they are not protected. Okay, so it squeezes out. This is how the excitation takes place. This process is called as a excitation process. For 2 marks they may ask you what is the excitation process. So metacyst enters the body, and then in stomach nothing happens, it is protected. In the small intestine nothing happens, it is protected. At the end, when it comes towards the end of the small intestine, at that time the excitation process starts and make the perforation in the cell wall or cyst wall rather, and from that small perforation, they make the perforation. From the small perforation they squeeze out, they comes out of the cyst wall, and now initially it is having 4 nucleus. Now in next stage what will happen? Nuclear cell division takes place. Okay, so 4 nuclear division takes place, only nuclear division, remember, not the cell division. Initially the cell division doesn't take place. The 4 nuclei divided into 8 nucleated form. Okay, and once these nuclei with some cytoplasm separate away from the parental body. So one by one all these nuclei separate. Initially the nuclear formation will take place, and from there a small cytoplasm, each nucleus will have some small cytoplasm, and from there onwards they will or they will try to go away from the parental body, as shown in the figure 3, number 3, as you can see, nuclear and cell multiplication. So metacystic trophozoite. Now trophozoite formation has occurred. So these are the young trophozoite. Now it passed through the ileocecal wall into the neutral or slightly alkaline environment of the large intestine. So it passed through the from small intestine towards the large intestine, that is the cecum and colon, which is the actual area of habitation. Okay, now sometimes under certain conditions some of the dividing these minuta, okay, this is the minuta form, trophozoite form, which has passed into the large intestine. Some of them sometimes cause the disease or disease forming part, that is the magna. So these minuta get converted into magna. This is also possible. Okay, now mature minuta form trophozoite. Before this magna formation, their number increases mitotically. So trophozoite reproduce mitotically by constant binary fission, in which 6 chromosomes are formed. Okay, 6 chromosomes are formed, replicated and shared by the daughters. So again 6 chromosomes are formed, again replication takes place, again the daughter formation will be there, and this is how the continuous mitotic, their number increases like this. Okay, so these lumen dwelling trophozoites, these are not pathogenic. So again I repeat, minuta form is not pathogenic but have a commensalistic deposition. It will remain in the colon region as long as it is there. It will develop, number increases, no problem, because it is feeding on the remaining food material or on the bacteria, not on the tissue. Now once from minuta form, magna formation is there. Okay, here onwards you have actual disease infection. So as you can see, initially cysted form, cyst enter into the body through water or any food contamination, from stomach towards the small intestine, at the end of small intestine, and initial it changes, it comes out of the cyst, 4 nuclei are there, and from there onwards there will be division of 8 nuclei formation, and from after the division of nuclei, single nuclei will separate out, and they are the cyst, they are the form. Okay, so this is the minuta form. Now magna forms. Now once the magna formation takes place, they will again have the binary fission. During the binary fission itself, there is a possibility of magna formation. Now this magna, what they do, the magna, it enters or it goes towards the intestinal part. Now what is this area? The magna secrete a collagen-lytic, okay, proteinase. These are the enzymes which digest the collagen, okay, or you can say the proteinase are the enzymes which digest the proteins, and there are some collagen is made up of protein. So these enzymes basically are called as collagen-lytic enzymes, also proteinase, which hydrolyzes, cuts the structural intercellular tissue proteins of the intestinal mucosa. This part is a intestinal mucosa and submucosa, okay, enabling these particular pathogens to enter into the tissue, penetrate between the epithelial cells. So we have a layer of epithelial cell, as you can see, this is the layer of epithelial cells. They cannot enter directly. How they can enter? After secretion of proteolytic enzymes, they will make or digest the collagen, they will start digesting the tissue, and they will open this area so that they can enter here. Now trophozoite grow at the expense of host tissue. Now they are not eating or they are not dependent on the digestive or pre-digested food or bacteria which is present in this colon region. No, they are not. Now they want to feed on the human tissues. Okay, this is one diagram. As you can see, this diagram is shows that if excess of these pathogens are present in the intestine, what they do, they will damage the intestinal layer, and as you can see, because of this damage everything comes out. So they may damage this. Now here what happens? Magna, what happens here? They grows at the expense of host tissue and lay a trail of destruction in their way, bringing about the necrosis, that is a localized death. So here localized death may also occur because the dead tissues are present and they penetrate deeper and deeper. So they go on, they go on, enter here, they will transfer through all this, the intestinal wall, they will eat through the intestinal wall, and what happens? Multiply at the during the same time they start multiplying by binary fission. Okay, this is also useful for penetration of this species. So binary fission will be there, their number will increase, they will start penetrating, they will create the hole itself and goes on increasing in the number. So this life cycle recommences only if the new host ingest, right? So the infection, this can produce only by the minuta forms, not by this particular form, but in some condition they transfer themselves. Okay, so magna trophozoite of Hystolytica may be considered as a biological dead end for this species. The only thing is they increase the number. But here also what they do, there is one more thing, the conditions favoring production of these trophozoites and their tissue invasion activity include the may include diet and nutritional status of the host. So intestinal bacterial flora, depletion of the mucosa covering the intestinal epithelial cell, concurrent with other parasite, decreased immunity of the host can be responsible for this thing. For example, a high protein diet of the host is generally unfavorable for the parasite. Okay. On the other hand, presence of certain bacteria such as E. coli, Escherichia coli in host intestinal facility, the establishment of magna forms and promotes invasion of host tissue. Okay, probably the bacteria utilizes oxygen in the intestinal lumen, and the anaerobic environment enables the parasite to perform normally and invade the tissue. So invasive amoebic infection does not develop in the intestine rendered free of bacteria by antibiotic. Invasion and establishment of the magna forms in the intestinal mucosa is achieved as the under suitable environmental conditions. Okay, so parasites make an intimate transient cytolethal contact with the several host tissues in the area where the intestinal mucus appears depleted and the host cells plebs or the round up and are rapidly ingested. Also, I should tell you one thing, that we have talked about the mitochondria. Less mitochondria or less number of mitochondria is present. So why it is so? Because it's a total anaerobic condition when they are inside, and glycogen is present. So basically, instead of instead of use of mitochondria, where you have you require oxygen for proper ATP development, instead of that it will go to the it will go for the anaerobic development of ATP, that is glycolysis only. Okay, so they don't require it, that's why the mitochondria is not present. Okay, so previously I have told you that in some conditions mitochondria is not there. The reason is this. Okay, so so the transmission, only 4 nucleated metacysts infective when evacuated by the way of feces. They may survive outside of the host body for about a month if conditions are suitable. Only with the food and water contamination by night soil containing ingested by person, parasite can resume their life cycle. Trophozoites extremely delicate, they may survive outside the host body only for 2 hours. Remember, without cyst wall they cannot survive, they require cyst wall. Okay, so in all these stages, as you can see, cyst wall is very important. Even if they are ingested with this period, they are killed by the host gastric juices. Without cyst wall they cannot infect. So transmission from one host to another can therefore occur only by the way of metacyst. Remember, this is only the possibility. Now, how transmission, how it takes place? The worst offenders in this context are the symptomless carriers such as food handlers, cooks and hotel boys. So generally we say that don't eat outside, or whether there is a cleanliness or not. Why we say that you should not eat from the open areas or open hotels? The reason is, the person who is handling the food may have these cysts. Okay, with as they don't wash their hands. Many times we have seen people give you water and their fingers are dipped in that water, and that they give you. So there is a possibility they have nails in that, there is a possibility of this metacyst presence. It will be there in the food or in the water, and when we drink it we will have the transmission. So transmission can occur when houseflies also responsible. So when houseflies or cockroaches visit the infected fecal matter and then unprotected food subsequently eaten unknown by the person. So yes, houseflies are also responsible for this, very important. And living metacyst may also be found in the excreta of these insects also sometimes. So these are all responsible for the transmission from one host to another. Okay, so I'll just repeat this part, that first of all it will be ingested, then in the intestinal area, incestation of small intestine, there from it will come out. From there division will take place 4 to 8, then the formation of minuta. It will present in the large intestine. Now from large intestine, after some period when their number is increasing, at that time they will go towards the cyst formation. So pre-cyst formation will be there, cyst formation will be there, metacyst formation will be there. Okay, and here it will be passed into the fecal matter, and from fecal matter it may again digested because of some contamination. Now one more thing, during this life cycle itself a binary fission takes place. During this binary fission, minuta may get converted into magna. Okay, and these magna it start infestation or start eating the tissues of the large intestine. It will open up, they may enter into the blood, go towards the liver or muscles or brain and start eating it. Only thing is their number will increase. Okay, infection they cannot, by this way they cannot infect other person unless and until there is a blood transfusion. Remember, the main thing of infection is the metacyst and through the ingestion of food. Okay, otherwise this part up to this region they are non-lethal, but once they are converted into magna form they become lethal, and this will be the amoebic dysentery, or the person will have a very harmful effect on its body. Just think over this, that if the intestine pore formation takes place in the large intestine, that the food will comes out of and mixed with the blood, and that will be a very dangerous thing that can happen. Okay, so that's why food habitats should be a good one. Now what are the adaptations for the parasitic life? So what are the adaptation that has happened for such kind of life cycle that you should see? So here first thing, it is divided into two parts, vegetative and reproductive adaptations. Vegetative adaptations concerned with the life of the parasite within the host body. So what happens in the, what kind of changes it has done within the host. So first, absence of mitochondria, as I have told you. Medium in which parasites thrive is poor in oxygen. Just now I have told you, they have dispensed with the metabolic process. Okay, for example, oxidative phosphorylation will not occur because for that lot of oxygen will be required, and in the mitochondria lot of oxygen will be required, which so this in mitochondria oxidative phosphorylation takes place, which need oxygen and mitochondrial enzymes. So instead of that they will go for the glycogen itself, degradation or glycolysis. They will go for the glycolysis where you don't require a large amount of oxygen. That is an anaerobic ATP synthesis. Now they do utilize glucose and glycogen for energy production. So these can be degraded anaerobically. So this is the first adaptation that parasitic life can have. Second adaptation is the presence of contractile vacuoles. So these organelles required by most of the free living protozoans for osmoregulation is not necessary for Hystolytica, since the parasite is osmotic balance. So contractile vacuoles, we don't see contractile vacuoles in these kind of organisms. The reason is parasite is in osmotic balance with its intestinal and extra-intestinal environment. So it doesn't require a contractile vacuole to maintain the osmotic balance, because the organism itself is in the osmotic balance, no need of it. Then the next one is the nutrition. So ability to secrete strong proteinase, okay, enables the parasite to invade host tissue. Even we have seen that it produces the trypsin for coming out of the cyst wall. Now trypsin can also digest the collagen. So these strong proteinases invades the host tissue. This is responsible for, or it is helpful in invasion within the host tissue and obtain the required environments. So an ancestor stage utilizes the stored glycogen and RNP as it matures. And the last one, encystation itself is a very good adaptation for infection purpose. So this state enables the animal to tide over unfavorable environmental condition outside the host body. So it comes out of the host, encysted form, it will remain without the moisture for 1 month at least, and during that period they will be carried by number of hosts. As I have told you, that the flies can do it, and cockroaches can actually do it, and if it is nearby the water body, then within the water body it definitely survives. So Entamoeba therefore become an obligate anaerobe, and when transmitted to the new host, the protective cyst wall enables the parasite. Even this protective cyst wall enables the parasite to safely reach its definitive site. Okay, so it protects from the stomach gastric juices and from the enzymes which are present in small intestine. Only thing why they are present in long intestine. The reason is there in long intestine no enzymatic activity takes place. That's why that is their main area where they are present. Now we will talk about the second part, that is the reproductive adaptation. Now in reproductive adaptation, the life cycle within and outside the host, what they do, rapid and constant binary fusion. Once they are in the host tissue, okay, they start the binary fusion. Once they are within the colon region, when they are not that much lethal, there also their number is constantly increasing. Okay, so nuclear and cell division, multiple fusion. So how this division is taking place actually? Nucleus, 4 nucleus was there, 8 nuclear formation, and suddenly 8 cells formation from single cell formation takes place. Now following encystation produces 8 amoebae from the single metacyst. So this increases the number rapidly. Production of very large number of metacysts, up to 5 crores in single discharge of the fecal matter. See, very important. Their number itself ensures that they will survive. Their number itself ensures that they will survive outside also. So up to 5 crores are discharged in single fecal matter of the infected person. So it maximizes the chances of least some metacysts. Out of 5 crore, at least 10 or 20 or 50 metacysts reaching a new host possibility is there. So if number is increased, more possibility that it will infect. So many nematocysts are usually inserted in given times. Reproduce success ensures, this reproductive success ensures the presence of large trophozoite population for the colonizing host tissue. Now both of these mechanisms ensures that the parasite is not exterminated by the host defensive reaction. So whatever, each and every host have its own defensive reaction, but because of all these adaptations, it ensures that parasite is not killed by the host. Although the high rate of the reproduction is seen in Entamoeba, in many other parasites, the strict sense it is not parasitic adaptation actually, but this number, large number, actually protects them. Okay, so this was about the adaptations and the life cycle. In next lecture we will talk about effects on the host and what are the remedies how to protect from the Entamoeba Hystolytica. Thank you.